Hi, I'm Seidi from Harvard Medical School. In this poster, we introduce Cistrome Explorer, a web-based interactive visualization tool for exploring epigenomics data with associated metadata. The goal of our work is to help analysts explore the compendium of epigenomics data in a flexible and scalable way. Domain-specific questions we aim to help answer include what transcription factors bind to cis-regulatory elements of interest and in which tissues. To support answering these questions, it is not only important to support scalable visual exploration with large genomic data and samples, but also to bring associated metadata such as tissue types and cell types to the analysis process. To address those problems, we developed Cistrome Explorer, a scalable, interactive, and generalizable visualization tool for exploring epigenomics data. To support scalable visual exploration of genomic data, Cistrome Explorer is built around HiGlass. HiGlass is an existing visualization library for exploring large genomic data sets with heat maps or genome browser tracks. This visualization scales to datasets with billions of entries by taking a data tiling approach. This visualization also supports useful interactions such as smooth zooming and panning using mouse scrolling and dragging interactions. We also integrated Cistrome Explorer with Cistrome DB, which is a resource that ingests thousands of publicly available human and mouse ChIP-seq datasets. By integrating this database, we are able to show diverse metadata for each sample and allow querying for additional information in our tool, for example, to find a list of transcription factors binding a certain genomic interval. The interface of Cistrome Explorer contains two main parts. In the middle, HiGlass visualizes epigenomics data such as peak values of ChIP-seq data. The x-axis represents the genomic coordinate, and each row of the heat map represents each Cistrome DB sample. On the left and right side of the HiGlass, we provide dedicated visualizations for associated metadata of each sample using bar charts and dendrograms. Here we are showing quality control values in bell chart, hierarchical clustering results in dendrogram, and cell types and tissue types in stacked bars. This is how it looks with more than 200 samples of ChIP-seq experiment dataset. Using the HiGlass heat map, you can easily zoom and pan the visualization to find interesting patterns or search for a certain gene. We provide further interactions in the metadata visualization for flexible exploration. Using the metadata shown on the left and right side, you can rearrange the rows of the visualization. This can be useful when you want to see consistent or different heat map patterns, for example based on tissue types or quality scores of each sample. We also support highlighting and filtering to enable focused analysis. For example, you can filter out samples with low quality scores. Our tool also allows a simple overview plus detail view by selecting a subset of rows with metadata and adding them as a separate tracks on the top or bottom. For more in-depth data exploration, we support making a query to Cistrome DB toolkit. For example, you can select the genomic interval and search for transcription factors that are likely to bind in the selected region. We support 3 different queries from Cistrome DB toolkit and allow users to easily make different queries by changing parameters and look into the data table of each request at any time. We are working on making this table interactive to support updating the visualizations based on the result for iterative analysis. As future work, we are considering 2 main aspects. First is about the generalization. We are currently allowing users to use a simple declarative grammar for arranging visualizations. In addition to visualization designs, we are planning to support using any kind of genomic data, not limited to the Cistrome data. We are working to document a file format that stores multiple big samples with associated metadata at different resolutions with the HiGlass core team. Secondly, we are actively working on supporting analysis of large number of samples. The current filtering interaction can be helpful for focusing on the subset of samples, and we are also implementing different aggregation approaches, such as allowing users to interactively aggregate rows with the same tissue types. We are testing our approaches with up to 10,000 samples from Cistrome DB and planning to support the improved scalability in the near future. Lastly, to support storing large data in a more convenient way, we are working on server-side support for scalable object store systems such as AWS S3. In conclusion, we presented the Cistrome Explorer, an interactive, scalable, and generalizable visualization framework for exploring genome-wide data with associated metadata. It currently supports different visualizations for metadata such as bar charts and dendrogram with different interactions. We are planning to support a generalizable visualization library for using general genomic data and supporting large sample sizes. Thank you for watching. Feel free to contact us with any questions.